I'm Tom O'Hare with Covaris. I'm presenting today in place of Deb. Unfortunately, couldn't be here. And then Dr. Jessica Chapman will present on her work from Memorial Sloan Kettering. Our sample prep offerings are built on our patented AFA technology. Using a patented technology, we are able to use a highly controlled, temperature-controlled, high-frequency ultrasonic air system. We'll provide highly controlled cavitation, which can shear DNA and lyse cells with no sample contact. The ultra-high frequency electronics and specialized transducer enable the acoustic energy to be focused within a sample vessel immersed in a water bath. The acoustic energy forms cavitation bubbles. These AFA-generated cavitation bubbles grow to a critical size, as you can see on this slide here, and that building collapse of the bubbles is what shears DNA and lyses cells in a highly controlled process. AFA has been the gold standard for shearing of DNA in almost every NGS lab, and AFA is used in many genomic and epigenomic-based applications, largely due to genomic analysis methods being well established for some time now, and the high-quality data produced using Covaris in those workflows. While proteomics and metabolomics are currently evolving at rapid speed. However, in all systems biology applications, the matrices are complex, and confident sample prep is critical for providing high-quality data. Over the years, AFA has been providing solutions for obtaining confident data across a host of different application types, from monitoring biomarkers to bottom-up analysis of proteomes to addressing complex samples such as FFPE. Once again, AFA is a non-contact, temperature-controlled sonication technology that can be scaled based on throughput needs and can be optimized to address the critical application requirements. Enabled by AFA, we have developed a one-plate sample prep workflow that can improve multiple steps of sample prep for protein analysis, from tissue disruption, cell lysis, to enhancing immunodepletion processes, to expediting digest without compromising data quality. This one-plate sample prep option has been a game changer for several proteomics laboratories. Advancements in the world of LC-MS have made single-cell proteomics a reality. However, the starting amount and yield can still impact the quality of data. High-quality sample prep is needed to ensure highest analyte recovery. A versatile workflow is super essential for any single-cell analysis from genomic and epigenomic to proteomic. This can be made possible with AFA. This slide is an example of what can happen with and without AFA. The goal was to identify and quantify 130 plus biomarkers, while the traditional approach on this side here was barely able to enable detection. And when we added the AFA as an enhancement, you could see that you got much higher identification and quantification. For TMT-based analysis, not only can AFA enable effective tagging of proteins, the entire process may become significantly simpler with AFA. In this particular case, a traditional process that takes 40 plus hours, as seen on the top here, and 50 steps can now happen in less than 25 steps and in 8 hours, which still ensures highest quality data. And you can see there's a large difference here between the top and bottom. Those are just two examples, but there are several other protein-based applications that can benefit tremendously from AFA-powered one-step sample prep. From monitoring phosphorylation to tracking PTMs, from purification of proteins to hydrolysis of protein cells, every application can benefit from the usage of AFA. This slide here just shows how a strong record of publications we have by labs utilizing AFA in their workflows. There's just a small sampling of the publications that reference both Covaris and AFA. Covaris has a full line of instruments, consumables, and reagents, which can assist with processing from single sample all the way up to 384 well plates. And this helps address the different throughput needs of the labs, as well as different sample sizes. We're a privately held company with placement of over 6,000 instruments at this point, and we've worked with several labs across genomics, epigenomics, proteomics, as I previously mentioned. At this point, I want to thank you for your time and let you know if you have any questions. Jay and I will be available in our booth 507. And at this point, we're thrilled to have Dr. Jessica Chapman come up and talk and present on her research and work she's been doing at MSK. Thank you.
Sorry. There we go. Thanks, Tom, and the Kavaris team, and thank you for you guys coming this morning towards the end of the conference. I know it's a little harder to be up early before the main program. Okay, so I'm just going to talk about some of the applications and pre-analytical workflows that we've been working on optimizing in collaboration with Kavaris, utilizing an Elite 220 RSC system that we have in our lab. So to give you some context, I am at Memorial Sloan Kettering Cancer Center, and so we're both a hospital and a research institution, but I sit within the hospital and am the director of a clinical proteomics laboratory that is part of the hematopathology service within a clinical pathology and laboratory medicine department. So we are the department that deals with patient testing for diagnostic purposes as well as prognostic purposes. We're regulated within the United States. Different states have different regulatory bodies. We're in New York State, so we're regulated by the Department of Health of New York State. We are a CLIA lab, and all the folks that work in my lab who do clinical work are required to be licensed to perform clinical laboratory science by New York State. Just give a context of how we're kind of operating and viewing these pre-analytical workflows. So when we think about pathology, we think of the study of the causes of disease and effects of disease or injury, and we think of this as a bridge between science and medicine. And in our setting in the hospital, we really are that connection between our clinical colleagues and then the science and development and implementing that for improving care of patients. And primarily when we think of pathology, we think of a clinical pathologist sitting at a microscope. But in a modern pathology department, we're actually using very advanced technology. Cytogenetics has been around a long time, but is very important in characterization of disease, especially in cancer. We have a very robust flow cytometry laboratory that looks at cell surface markers and identifying abnormal cell populations and characterizing those. Memorial is also very well known for our molecular pathology service, which has looks at mutation profiling in order to characterize disease and get patients the best treatment possible. And so the clinical proteomics laboratory is becoming part of that web of diagnostic tools. And so our goal is to take these clinical specimens that are kind of standard within the hospital for collection, how they're collected. We can't really change the whole process. So paraffin-embedded tissue, blood, serum, plasma, and then live cells, fresh cells, which we get either from our flow cytometry lab, and extracting protein from all of these different complex clinical specimen and utilizing primarily bottom-up mass spectrometry-based proteomic workflows and resulting in clinically relevant data. So whether that means data-dependent, targeted, or DIA depends upon the question, the assay. But our main issue is how do we extract the protein from these complex matrices in a way that is reproducible. So first I'll just talk about our work with Kavaris on optimizing an FFP workflow. So we know to get tissue to that state, it starts by, we get the biopsy, it gets fixed, it gets paraffin embedded, then we get thin slices cut around 4 to 6 microns, placed onto a slide and staining of those sections for characterization under microscopy. But that tissue is also used by our molecular colleagues, cytogenetics, and then we are looking to utilize that tissue as well. So many years ago, an assay was validated using FFP tissue in our lab, and this is the workflow that was validated. You can see that it takes about 16 to 24 hours. Maximum, we're processing 24 samples. You'll notice, just to point out, we don't have deep paraffinization on here because this assay is taking tissue from slides after deep paraffinization on the slide. So it's quite a long process. We do a two-step heating process to reverse cross-linking. Homogenization, we were just doing in a water bath sonicator in 0.5 ml tubes. So we wanted to work on utilizing our Covaris system for this. So we first looked at homogenization, and we tried replacing that homogenization where we just put these tubes in little floaters in the water bath and move that onto the AFA system. So benefits of that are that we can utilize a 96 well plate, and so that increases the throughput, but also means that it's easier to increase the volume of samples. So when we looked at that simple comparison there, we see that the amount of total protein by BCA assay that we extract is very comparable, if not slightly better, with AFA. So shown here is just taking healthy spleen sample, 6 micron section, scraping that into either a tube or the Covaris TPX plate wells, and this is in 50 microliters of a dilute zwitterionic buffer. The reason we use that buffer system is that what was validated years ago, and we just wanted to change one thing at a time. Next to that, you can see when we screened a small portion of these samples by data-dependent acquisition on our Q Exactive, protein group is very comparable, and then peptide group, there's a small difference there. And these are 4 replicates of the same tissue performed at the same time. Total overlap of proteins identified is nearly the same, as shown in the Venn diagram, so that's protein groups identified. We also pull out data for specific proteins of interest, in this case immunoglobulins, because that's part of what we're going to be looking at when we're looking at these plasma cell disorders I'll talk about in a bit. And so abundance of label-free, this is doing label-free quantitation from our data-dependent acquisition. We see pretty comparable overall abundance of the different immunoglobulin heavy chain constant regions as well as the light chains. So we put the AFA in place for our homogenization. We also, you may have noticed, had a very long reduction alkylation process for some reason. And we just did the test. I'm not going to show that data. You probably don't need to see that, but we have to validate everything. So we validated that the results were the same. Next step for us was wanting to really try to push this workflow to fit in a single day so the samples can go onto the instrument at the end of the day and we have data in the morning. And so that involved taking the digestion and moving that onto the AFA and doing an AFA-assisted digestion step. So we were doing overnight digestion because it was just easier to put them on to digest and come back in the morning and do C18 cleanup. But to compare that to AFA digestion, we took again this healthy spleen tissue sample. We have the same buffer, 50 microliters. This was all done in the TPX plate, and so all the homogenizations on the AFA system. We did an overnight at 37 degrees on a thermomixer, 3 hours at 37 degrees, and then an hour and 3 hours of AFA time. And so I'm just showing miscleavage. This is from doing data-dependent acquisition and searching, and this is percent miscleavage on peptide spectral matches from that result. And so we can see that overnight, obviously the lowest amount of miscleavage, but the 3-hour AFA is only a couple percent more miscleavage overall. And then we also spike into our samples in order to make sure that we have complete digestion or as complete as possible digestion. We spike in a digestion monitoring or control. We are using Digestif from Promise Proteomics, which is shown here, and that's the publication regarding that. This data is just from an early experiment we did when we first started using this digestion standard, where we took that spiked into spleen FFP, and this is just done 37 degrees on a thermomixer and tubes. So this isn't involving the AFA, but this was just showing that we picked pairs, of peptides that have varying degrees of difficulty of cleavage sites and monitored by PRM, the fully cleaved and the one miscleavage peptides over time. And so then we were able to narrow down a set of peptides to target in all of our samples as a digestion control step. So using that, we had spiked in digestive into those samples that we used for AFA digestion testing, and we see that we don't detect any of the miscleaved products at the 3-hour AFA. So we feel very confident utilizing that 3-hour digestion step for our FFP sample prep. So this is in the end, we went from about up to 24 hours total time, processing samples in a 0.5 mil tube, and a maximum of about 24 that we can fit on a thermomixer at a time. And you can also imagine opening all those little tubes every time for each step. Move that onto a TPX plate, a 96 well plate, and condensing down that digestion time to a few hours so that we can get those, do our C18 cleanup and get those samples on the instrument. So we went from about 24 hours for 24 samples to roughly 7 to 8 hours, depending on C18 cleanup method, for up to 96 samples. And that's prior to getting on the mass spectrometer, put those on, and then go from there. So we were able to use the AFA to simplify this workflow, go from homogenization through to digestion in a single well. And this is compatible with automation. We don't currently have automation in our lab, but it is very compatible with that. And we can also, like this, because it's scalable for a clinical test, we don't know the volume we're gonna have in any week for certain tests. And so we can process a single sample, we can process a row of samples, 8 samples, we can process multiple rows or a whole plate using the same workflow. So I'll just show briefly, this is kind of a simple assay that we validated that's doing immunoglobulin relative abundance in FFP tissue. So I mentioned we're within the hematopathology service, so I primarily work on heme malignancies, but also solid tumors as well. So I'm going to talk about plasma cell neoplasms. And so we know that about 10% of all hematological malignancies are plasma cell neoplasms. And so these are diseases in which an abnormal plasma cell population is replicating very quickly, normally in your bone marrow and displacing the rest of those healthy plasma cells and healthy cells in the bone marrow. And so when we think of plasma cell neoplasms, most people would, multiple myeloma is the most common to think of. And so these plasma cells normally are fighting against infection. They produce monoclonal or clonotypic immunoglobulins. But in the neoplastic plasma cells, they're producing a monoclonal immunoglobulin, but they're producing a lot of it. And most cases, most multiple myeloma, they're secreting those immunoglobulins, and those are circulating out into the circulatory system. So just to give you an idea, this will be important as we go along. So like I said, this neoplastic population of plasma cells is producing this monoclonal antibody. And part of the process of diagnosis of a plasma cell neoplasm, first and foremost, is morphological review of a bone marrow biopsy. So this is a needle biopsy. These are about maybe a centimeter long, probably shorter, a few millimeters wide. So these are needle biopsies. And so the pathologist is going to look for these abnormal plasma cells in the bone marrow. Then they're going to characterize that immunoglobulin that neoplastic population is producing by immunohistochemistry or in situ hybridization. So they're trying to figure out if that is an IgG lambda, an IgA kappa, et cetera. So determining heavy chain and light chain subtype. However, those assays tend to have very high staining background, are difficult to interpret in a quantitative manner when comparing. Also, you're going to have a different section per stain. As you cut through a tissue, the morphology changes, so it complicates this. Sorry. There's also not really standardized protocols for some of these assays. So we were asked to develop and validate an FFP tissue, an assay that would determine the relative quantitation of the different immunoglobulin subtypes in a bone marrow biopsy at a diagnostic time point. And so we have a Q Executive, and we developed a PRM-based assay for that. So we followed kind of standard protocol. We looked at theoretical digests of these constant regions for the IgG, IgA, IgM, IgD, and IgE, even though those are more rare, kappa and lambda constant regions. We looked at what peptides should be unique. We purchased recombinant proteins, we digested those, we spiked those into matrix, and then we went about doing our limit of detection, limit of quantitation studies, et cetera. Then we apply that to a selection of 44 bone marrow biopsies. So the idea is we do PRM, we use Skyline to quantify, and we have an output that shows relative percentile of those different immunoglobulins. So sometimes it's very obvious and sometimes it's not, but so the idea would be that we then look at the results from that assay, and we compare that to the immunohistochemistry results, because these are all biopsies that have been characterized. So in this case, on the top circled in green, I'm showing if there's known results by immunohistochemistry. This is a pretty obvious one, in that the lambda immunohistochemistry stain is quite dark and the kappa is pretty much negative. And we can see that aligns with the results we get by mass spectrometry. Nothing too fancy there. We also compared it to standard serological testing that's performed on these patients that just determines if they have in circulating I'm not going to use the pointer over my shoulder, but if they have a monoclonal immunoglobulin in their serum. So the first plot is showing in green a healthy patient. That's what your serum protein electrophoresis should look like. If you have a plasma cell neoplasm, you would have this red, in the red trace where you have this big spike where you have this one single monoclonal immunoglobulin. And then immunofixation is where you take that and you use anti-sera to determine the subtype of that immunoglobulin. So in this case, it's showing an IgG lambda. So I'm just showing kind of, these are standard testing in a clinical lab. And then we compare our results to those results as well, because all of that should align. So our validation set was 44 FFP biopsies, bone marrow biopsies that from cases that were diagnosed as a plasma cell neoplasm. They have to have 30% or more of the disease burden as determined by a CD138 immunohistochemistry stain. So we wanted to apply this workflow for processing to a subset of those tissues because we'd already done the validation using a more conventional or previous workflow. So we took a subset of 15 of those, and you can't really read this, so don't worry, and then myometrium control. And I'm just showing here, we put the results side by side. So the relative abundance values might vary slightly, but the overall results are concordant, meaning that we would call it an IgG lambda. It met the requirements we had set through the validation. We'd call it an IgA kappa. And so this is just showing one case example, just showing that this, the results between processing FFP tissue both ways, the results were the same. So we showed that we were able to implement this pre-analytical workflow into our assay. And able to do this more quickly, and since this is potentially a more a higher volume test, that then we can put this on the AFA and keep up with demand. And that can decrease the turnaround time as well, which will be important in the clinical diagnostic setting. So we are waiting for approval from New York State of our laboratory developed test, which is a long process, but then we will implement this assay utilizing the AFA workflow. So I want to transition to talk about how we're using the AFA for a different specimen type. So we have been working for a while with Covaris on trying to utilize the technology for different specimen type because we don't have a lot of lab space. We're in a very crowded city, and we just keep rearranging our little lab space to accommodate upgrades of equipment and new things we need. So we want to use this one instrument to do as much as we possibly can. So, as I mentioned before, we have a very strong flow cytometry lab. And so there is some workflows we want to implement where we would get sorted cells, where we get like an enriched population of neoplastic cells from our flow cytometry lab. We also have some workflows where we would do a bead-based sorting of cells. And we want to take those fresh cells and we want to lyse them, process them as quickly as possible in order to get those to our analytical analysis. So this is just an overview of the assay or the pre-analytical workflow that we've worked out for processing our cells. And so we utilize AFA for lysis, and then we do our reduction alkylation, and then we do SP3 cleanup because we can keep everything in those wells and do it in that plate. And so we do our SP3 binding with AFA and washing with AFA, and then we do digestion. In this case, we have that protein bound to those beads for the SP3 cleanup, and we're doing the digestion in the AFA on the beads. So I'll walk through a little bit of that. So this work that we did, we've optimized initially the optimization work. We used 50,000 white blood cells. So we just take blood that we get from our rapid response lab. We do red blood cell lysis. We have a white cell pellet. We resuspend that into our lysis buffer. We're using a 1% SDS and 100 millimolar ammonium bicarbonate. We create a suspension and then we transfer aliquots of that into the plate for these initial optimization. And it's a 20 microliter total volume that we're processing the samples in. So this is just showing the SP3. We'll come back to this as well, but we've taken this workflow. We can do this in about 6 hours for a full plate, and somewhere around that a little bit less if we're processing less than a full plate. So I'm not going to spend any time comparing. We did a lot of optimization of our homogenization, our lysis, I'm sorry, and if you're interested in those settings, definitely talk to myself or Tom and his team because they have a lot of really helpful resources there. But I want to talk about AFA-assisted digestion because I think that's where this workflow really is beneficial in that we don't have to put on our digest to sit there overnight or even for 6 hours to get a good completion of digestion. So this is just showing we did those 50,000 white cells in 20 microliters. We did 4 replicates per condition. And we did an ON is overnight digestion, which is just 37 degrees on taking that plate, putting it on a thermomixer for 37 degrees overnight. And this is all with SP3, so the proteins are bound to those SP3 beads. And the trypsin, we're doing it, actually, it's a pretty high amount. We're doing a 1 to 10 because we wanted to keep it consistent. And for the short AFA, we're using a significant amount of trypsin there. So the AFA 60 is 60 minutes, 120 minutes, and 180 minutes. So this is just screening an aliquot of that data dependent on the Q-exactive. And we can see that our protein groups are pretty comparable across those when we load the same amount of material, similar with the peptide groups and on there. But what we are most interested in evaluating the AFA-based digestion is miscleavage. So on the left in the dark blue, making sure my left and right are correct, is the percent miscleavage PSMs for doing a search against a uniprot human database. And so we can see that as we increase the amount of time of the AFA, of course, we get fewer miscleavage. We're not quite at overnight levels with the SP3 bound digestion. And the light blue is using that digestive standard. So we do still see at the 120 and the 180 a little bit, but we're talking about less than 1% of that peptide in the miscleaved form compared to the fully cleaved those peptides that we're monitoring. So based on this, and because that SP3 binding does add time onto our processing, we decided that we're actually going to move forward doing a 2-hour AFA digestion for our cell lysis work, because it seems to be working well with majority of protein biomarkers that we're looking at. So we actually cut that down to 2 hours just because of the total time it takes for processing, and because we want to get those samples on the instrument before the end of a day. And because we're working in a clinical lab, that end of day is pretty firm there. So we kind of have to fit it in there. So what we've been working on now, because we're going to be implementing this on lower level samples, and we're repeating these experiments to increase the reproducibility at these lower level, what this is showing is we decided we diluted down our cell suspension. And so we have examples here where we did AFA, assisted digestion, for 2 hours versus overnight digestion at 37 degrees with 50,000 cells, 10, 5, and 1,000 cells in that 20 microliter volume. So all of the other conditions are the same. We just did a dilution of the cells. And so you can see this is screening by data-dependent acquisition, again on the Q-Exactive. And we can see that the protein groups identified in this process are actually a little bit better when we're using our AFA digestion. And then peptide groups also similar. We can see that some of those error bars are quite large in the lower number of cells. So we're repeating that, and we're also looking at trying to get some kind of automation going on there for this processing, just because with using the multi-channel pipetter is not always as reproducible depending on who's processing it. And we need reproducibility for a clinical setting. So this is basically where our workflow, I apologize there, it still says 3 hours on the AFA. So now we are looking at also applying this similar workflow to some fine needle aspirate samples from lymph node and lung, looking at small cell lung cancer. We're looking at this with urine samples that have been banked in order to determine if we can utilize this for assisting with diagnosis or prognostic evaluation for urothelial carcinoma. There's a lot of data out there about urothelial carcinoma and proteomics, and we're just trying to see what biomarkers we could reproducibly utilize truly to help with diagnostics. But we're utilizing a very similar workflow for all of those types of specimen, because the starting point is pretty much the same. We have some kind of material, we want to extract the protein, we know we can do it reproducibly using this system, and then also we can do this faster digestion and get those samples onto the instrument. So overall, we found that utilizing AFA has facilitated a simplification of our workflows, as well as development of simplified workflows for lysis. We can go for FFP homogenization all the way through, and then we are doing C18 cleanup on those. But we very recently, just a number of weeks, have had an EvoSep installed, and so now we're going to be hopefully just loading our samples onto the Evo tips and avoiding our separate C18 cleanup. And we also, with our cell lysis, we're going lysis all the way through to pulling those samples off the SP3 beads post-digestion and running those right away. And so this is also highly compatible with automation and doing high throughput workflows, which we aren't doing automation now, but we're trying to see if that would be possible, especially with EvoSep putting out an updated automated loading protocol. And so we've been able to move some of these workflows to single day processing, which is very important. And so we've been able to do this fast. It has scalability, which is important for us because we have variable numbers of specimen for these different tests at different times. And we've been able to see pretty good results with a variety of different true clinical sample types. So I maybe went a little fast, but I just wanted to acknowledge my team. The Clinical Proteomics Laboratory, and we are small but scrappy in hematopathology service for kind of providing resources so that we can get this, keep moving forward in our endeavors. and then the team at Covaris, including Tom and his colleagues, who we do a lot of kind of troubleshooting together on things, and they've been a great resource in getting this up and running, and the funding source that specifically supports our diagnostic development. And if you have any questions or comments. Does anybody have any questions? Hi.
Hey Jessica, really nice talk. Have you tried adjusting your temperature for your trypsin digestions?
In like on the thermomixer type or?
Yeah, on the thermomixer with the APA assisted.
So on the AFA, we have the temperature set. It's about 30 degrees when we set. There's a water bath in the AFA system. We haven't played much with the temperature on the AFA digestion, and we have not on the thermomixer either. Do you have experience with doing a?
Yeah, I've seen some research and protocols do it quicker at 42 degrees.
Okay.
But I don't have personal experience in this quicker, but I have seen that before.
No, it'd be interesting to see one of the benefits with the AFA as opposed to when we did the water bath, like homogenization, obviously, like your temperature varies, right? So you have tube floating over here and it's not hot, and the one in the middle gets hot and things like that. So one of the benefits of the AFA is you shouldn't be getting temperature buildup. So it would be interesting maybe to play with the water bath temperature and see if that improves a little bit. We can keep that 2 hour and kind of get that extra benefit. Thank you.
Thank you. Actually, I really understand the clinical lab, high throughput scaling up is one big thing. But I'm curious about your comment on reproducibility and also the dynamic range of this method compared to like conventional ways.
Yeah. So our reproducibility with our like FFP workflow and our cell lysis, when we're at like 50 and even like 10,000 cell suspension, it looks pretty good. I think the lower cell suspension, the reason is we're just kind of diluting that. And what we want to get is some like real samples that are at lower cell numbers. So it's not like us diluting something. So I think part of our reproducibility with like total protein and protein groups has more to do with the human error we're adding in through our mock samples, because we're just like kind of diluting it. So we're trying to work with our flow lab to see if we can get some sorted like populations that are certain cell numbers. So it's more accurate determination of those cell numbers so that we can see the reproducibility of processing. We've looked at 20 microliters is low, but when we're getting down to low cell numbers, is that still too much volume a little bit? So we've been talking with some of those things with Covaris folks as well. Yeah, but.
Dynamic range?
As far as dynamic range of like the amount of protein or amount of input into the AFA system, or more like on a mass spectrometry side of
on your your endpoint mass spec data, like compared to conventional ways to do, like did you have difference in dynamic range?
No, I mean, when we're utilizing the mass spectrometry more as like kind of a screening evaluation tool, we're loading like the same amount of protein onto it, no matter what the way we're processing it. So when we're doing comparison, as far as complexity of the proteome we're extracting, it looks very comparable. We don't think we're like biasing when we're doing AFA versus something else.
Thank you.
I was trying to look at some. I also wanted to mention, just because these are questions that people have asked other times when I've talked to folks about this, we've been trying to do things in a lower volume. So our FFP, we're doing at 50 microliters. And then our cell lysis we're trying to do at 20. And then you can also go up to higher volumes if you want, depending. So some of our urine work, there's a way that the cytology lab processes those, and that we may be dealing with higher volumes. And so we are doing those in a more like 100 microliter type setting. And then we also, the FFP optimization work, we did not use microdissected tissue, but one of our clinical assays does use microdissected tissue. And right now we're not doing that with the AFA simply because our LMD scope stage is not compatible with a 96 well plate collection, but we received it. It's sitting in my office. I'm just waiting for installation, and then we're going to be moving that into the AFA as well because we're upgrading our laser microdissection scope to accommodate the plates, and so be doing the small amount, even smaller amounts of FFP tissue. So if anyone doesn't have any more questions, Tom, is there anything else you wanted to cover? All right. All right, thank you.